Formation of the ectoderm, mesoderm, and endoderm. The ectoderm, mesoderm, and endoderm are the three primary germ layers formed during gastrulation, a key developmental event that occurs after the fertilization of an egg. These germ layers give rise to all the tissues and organs of the body. Their formation is a critical process in the establishment of the basic body plan of the embryo. 1. Overview of germ layer formation. Gastrulation is the process through which the bilaminar disc, composed of the epiblast and hypoblast, transforms into a trilaminar disc. This process establishes the three germ layers: ectoderm, mesoderm, and endoderm. The ectoderm, mesoderm, and endoderm are the foundational layers from which all organs and tissues will develop. These layers are formed through complex cellular movements like invagination, involution, epiboly, and convergence extension. Second formation of the ectoderm. The ectoderm is the outermost germ layer. It is formed from the epiblast cells during gastrulation and is crucial for the development of the body's external structures. Development of the ectoderm. Epiblast cells at the surface of the developing embryo will remain in the ectodermal layer, while cells migrating inward during gastrulation will form the mesoderm and endoderm. The ectoderm will later give rise to skin and hair, epidermis, and hair follicles. Nervous system, brain, spinal cord, and peripheral nerves. Sensory organs, eyes, ears, teeth, enamel. Parts of the face, such as the oral cavity and parts of the jaw. Formation of the neural plate. Early in neurulation, a subset of ectodermal cells differentiates to form the neural plate, which will eventually fold and form the neural tube, giving rise to the central nervous system, brain, and spinal cord. Third, formation of the mesoderm. The mesoderm is the middle germ layer and gives rise to many important structures, including the musculoskeletal system, circulatory system, and kidneys. Development of the mesoderm. During gastrulation, cells from the epiblast ingress move inward through the primitive streak, forming the mesoderm between the ectoderm and endoderm. The mesoderm will later give rise to skeletal system, bones, cartilage, muscular system, skeletal, smooth, and cardiac muscles. Circulatory system, heart, blood vessels, and blood cells. Excretory system, kidneys, ureters. Reproductive system, gonads, reproductive ducts. Dermis of the skin, deeper layers of skin. Lymphatic system, including spleen and lymph nodes. Mesodermal development. The notochord, which forms part of the mesoderm, is essential for the patterning of surrounding tissues, especially the neural tube. It also plays a key role in the dorsal-ventral axis of the embryo. Fourth, formation of the endoderm. The endoderm is the innermost germ layer. It forms from the epiblast cells that ingress through the primitive streak and become the endoderm. This layer is primarily responsible for the formation of the internal organs. Development of the endoderm. As epiblast cells move inward during gastrulation, they form the endoderm at the deepest part of the embryo. The endoderm will later give rise to digestive tract, stomach, intestines, liver, pancreas. respiratory system, lungs, trachea, bronchi, endocrine glands, thyroid, parathyroid, thymus, urinary system, bladder, urethra, liver and pancreas from the foregut endoderm. 5. The role of signaling pathways in germ layer formation. The development of the ectoderm, mesoderm, and endoderm is regulated by various signaling pathways that control cell fate, migration, and differentiation. Key signaling pathways involved in germ layer formation. Nodal signaling. The nodal signaling pathway is essential for mesodermal and endodermal development. It regulates the formation of the primitive streak and is important for the induction of the mesoderm and endoderm during gastrulation. WNT signaling. WNT signaling is involved in anterior-posterior axis patterning and mesodermal patterning. It helps establish the neural ectoderm and regulates neural tube formation. BMP, bone morphogenetic protein. Signaling. BMP signaling is crucial for ectodermal patterning and for promoting non-neural ectoderm fates while inhibiting neural development. It plays a critical role in forming the epidermis from ectoderm. Hedgehog signaling. Hedgehog signaling is essential for notochord development and neural tube patterning, which is essential for proper mesodermal and ectodermal differentiation. FGF, fibroblast growth factor. Signaling. FGF signaling helps in mesodermal formation and promotes neural differentiation. It is involved in somatic formation and muscle differentiation from mesodermal cells. 6. Summary of germ layer formation. Gastrulation is the process that generates the 3 germ layers: ectoderm, mesoderm, and endoderm. These layers will differentiate to form all the tissues and organs of the body. Ectoderm forms external structures such as the skin, nervous system, and sensory organs. Mesoderm gives rise to structures such as muscles, bones, and the circulatory system. Endoderm forms the internal organs like the digestive and respiratory systems. Signaling pathways like nodal, WNT, and BMP control the differentiation of cells into the appropriate germ layers, guiding the development of the body plan.